Cloning in bacteria other than E. coli. So this is the next chapter that we are going to start. Now we have the idea that how DNA, it can be isolated, cloned, and how the expression can be detected, and what are the different techniques to detect the expression of RNA or protein molecules that we have discussed under the heading of functional genomics and proteomics. And now, before going to discuss the applications of recombinant DNA technology, we are going to have a brief look at how and which type of different organisms they can be used as a cloning host, and especially other than E. coli as far as microbial group is concerned. And here the first topic, it is cloning in gram-negative other than E. coli. About E. coli, we have discussed repeatedly that it is the host of choice because a lot of studies have been carried out by using this suitable host organism. But all the time, it is not feasible to use E. coli. Although variety of plasmid or carrier molecules are available for this group of bacteria. But some or all metabolic pathways, they are not present in this organism. Like if we want to study something related to sporulation or pathogenicity or the degradation of some aromatic compounds or some other biochemical pathways. So all these things, they are not available in a single microbe like the E. coli. So with reference to target metabolic pathway, we have to use sometime related organism. And for other organisms, the basic step must be followed. Basic steps include that there must be a method available for the transformation of the host organism. कि ये DNA को uptake करे, जिसको हम transformation कहते हैं. It may be mediated by conjugation, transduction, or maybe electroporation. इसके बाद next step है कि DNA uptake हो गया है, तो ये उसके अंदर maintain रहता है कि नहीं. That is stability of the recombinant molecule. Because sometimes it is not stable and it may be restricted by the host organism. And third step is that it must be expressed in the target organism. So all these points we have already discussed. तो हम यहाँ एक एग्जाम्पल कोट कर सकते हैं कि इन केस ऑफ Pseudomonas putida, ये एक ऑर्गेनिज्म है, माइक्रोब है. तो अगर हम कोई जनरल रिकॉम्बिनेंट मॉलिक्यूल इसके अंदर इंसर्ट करें, ट्रांसफॉर्म करें, तो वो स्टेबल नहीं है. सो नो ट्रांसफॉर्मेंट्स में बी डिटेक्टेड. बट if the transformants are recombinant molecules, first they are synthesized from another microbe like Pseudomonas tutzeri, that is closely related to Pseudomonas putida, then transformation or the replication, it is possible. So naturally, variety of microbes that may be up to 40 genera, they have the ability to naturally uptake the foreign DNA. And sometimes we have to develop competency, that is the ability to uptake the foreign DNA in the host organism. Now, at the end we can say that what type of gram-negative microbes other than E. coli, they can be used for cloning. So for this purpose, sometime we have to develop shuttle vector that have the ability to replicate in two different hosts, or a vector or a plasmid have to use that is having broad host range activity. Because if we will attempt to transform gram-negative microbes by using some natural plasmid, so desirable properties with reference to their small size, multiple cloning sites, selectable markers, they may not be available. So for this purpose, we have to use E. coli as an intermediate host by using a shuttle vector, or a broad host range plasmid. So such vectors, they must be there. So first, such vectors, they may be replicated within the E. coli, and then such recombinant molecule, they can be used to insert other gram-negative microbes. So here we can quote a few examples of such microbes like Thermus, genus Thermus, and then Desulfovibrio, and some other gram-negative microbes like Pseudomonas. They can be transformed by using the recombinant molecule that have been synthesized or replicated within the E. coli. So under this situation, it must contain selectable markers that not only going to express in the E. coli as well as in other gram-negative microbes.